Welcome back, friends. Welcome in another video tutorial. This will going to be a recombinant DNA technology video where we will going to discuss about what is the difference of transgenic animal and cloned animal. I find it quite interesting that it is very difficult to understand about what is the difference of transgenic animal and cloned animal. So today I will going to explain you. So try to remember that how we have created transgenic plant in the application of recombinant DNA technology. How we are using Agrobacterium tumefaciens to transfer our gene of interest, which will integrate with the host genome, right? And now when those cell divides into another two, that is the daughter cell will also have the same foreign gene, right? So we are calling them as transgenic plant, right? So remember it very carefully that the transgenic is a word that is only used for those animals or plant who has or had one or more gene from another cell or organism, that is the foreign gene for it, and stably incorporated into its own genome. Okay, not only that, it is capable of passing on this foreign gene into the daughter cell. Okay, so such kind of animal can be called as transgenic animal. Okay, that means they are having foreign gene which is integrated into their own genome, and they have the ability to pass those foreign gene into the daughter cells. Okay, therefore a transgenic organism, or let's say transgenic mouse for example, may have a foreign gene construct which is stably incorporated into its own DNA, okay, that ultimately causing the expression of that foreign gene in cell or tissue that may not otherwise be expressed in, right? So another important thing that here I want to mention, that the expression level of the foreign gene in the host organism should be different from the expression level of other gene in that host cell or organism. Okay, that means suppose you have created a transgenic mice which contain a foreign gene incorporated with its normal genome, right? So the genes which are present in the normal genome will be expressed at a level which should be different from the gene which is foreign. Okay, that means the expression level of the foreign gene should be different from the expression level of the normal genes. That is another important criteria. Okay, so we can create transgenic mice that have the ability to overexpress or underexpress a given gene to study the function of that gene. That means those kind of transgenic organisms are very useful to study the function of a gene. Okay, so that is all about the transgenic organism. Okay, so transgenic organism means organisms who have a special type of foreign gene. That foreign gene is integrated into its own genome, okay, and it have the ability to express it. Okay, not only that, it also have the ability to express, or it also have the ability to pass those foreign gene into the daughter cells. Okay, and the expression level of those foreign gene is quietly different from the expression level of the normal gene present on the host genome. Okay, clear? So now we will going to talk about knockout gene. What is the meaning of knockout organisms? We have, I think you may heard about knockout mice. So what is the meaning of those things? So remember it, knockout means when we are giving a foreign gene which is inactivated. Okay, that means organism having inactivated foreign gene will be referred to as knockout mice or knockout organism. Okay, and they are also very useful for studying the function of that gene. And now let's talk about the cloned animal. So what is the meaning of cloned animal? So try to remember, we have talked about cloning, right? So the cloned DNA should be identical with the DNA from which it is cloned, right? That is the idea. Also in case of cloned animal, that means a cloned animal will have the same genetic makeup or same will have a identical appearance with the organism from which it was derived. Okay, so suppose when we are doing gene library, right, so inside the cell our gene of interest is present, it will be divided, right? So this and this will be the cloned cell of this one. Okay, they are having same makeup with this one, right? So for example, a famous example was Dolly, a lamb cloned from an adult sheep udder cell. Okay, so that is the idea of transgenic organism. Transgenic organism means we have inserted gene of interest, right, which is integrated with its original genome. Okay, now it is expressing itself, right? That is why it shows some functions and we get the opportunity to study them, right? And transgenic organisms have the ability to pass those foreign gene into the upcoming organisms, right? Or that is their daughter organisms, right? That is daughter cells. Okay, and the expression level of the transgene will be different from the expression level of the normal gene. That is very important thing you should remember. And another important thing, what do you mean by knockout animals? Knockout animals means the animals who have a inactivated foreign gene. Okay, this is also very useful for studying the function of a gene. And last one is cloned animal. Cloned animal means the animal who have same makeup from which those are derived. Okay, for example, a famous example, Dolly, a lamb cloned from an adult sheep udder cell. So that is the difference of knockout, cloned animal and transgenic animal. I hope this video will be helpful for you.